I'd like to welcome you to today's research colloquium. I'm Eve Riskin. I'm faculty in electrical engineering and also associate dean in the College of Engineering. We're very happy to welcome Dr. Jacqueline Fairley. She is here from Emory University, where she's a postdoc in neurology. She has her PhD from Georgia Tech and went undergrad to the University of Missouri. She has been working in a number of areas relating to what we call big data or data science, applying in the medical arena. She has worked in particular at Emory on sleep science, and she has received a number of awards for her research, including grants from the National Institutes of Health and the National Science Foundation. So please welcome me in joining. So please join me in welcoming Dr. Fairley, who will be speaking about her research. Thank you.
Thank you for that lovely introduction. So before I begin, I just want to express my gratitude for having the opportunity to come to Seattle and speak with you all today. So thank you to the Electrical Engineering Department for hosting me. Just to get a feel for the audience, how many people are, I guess, engineers or electrical engineers in the audience? Kind of like a show of hands so I can, okay. And then, are there any individuals from, say, the biology field or medicine? Okay, all right, great. So majority of my talk will be heavy on the engineering side, but there will be some information on the biological side as well. So hopefully it's a great balance, so the people who don't have the experience in the biology side, they might find some interest in learning something they didn't know. And then the people that are interested in learning on the engineering side, I guess you'll get the best of both worlds, because hopefully you'll get a chance to learn a little bit more on the biology side and then also go a little deeper into some of the data mining algorithms that I'll discuss today. So the focus of today's talk is data mining the human sleep-wake cycle. And before I begin, I just wanted to say that the results that are presented here today was funded by the NIH F32 award. So just giving you an idea of what is the main goal with respect to this research. Well, in the area of sleep medicine, which is the area where I collect my data sets, they generally look at the data sets with respect to this visual analysis. So it's a classical approach of looking at the output from a sleep study and then making a decision with respect to the person's health. And, my contribution is to be able to not only make that in an automated way, but to also make it in such a way that we can learn even more things than the visual approaches. So that's why I have on there data mining. So we're taking the data and we're trying to, figure out what is the important aspects of it. Just to give you a little brief information about my background, Dr. Ruskin did a really great introduction. I just want to add a little bit of just my own personal experience with respect to my training. So I graduated from the University of Missouri, Columbia, and from that experience, I really was interested in learning a little bit more about research, because I did some undergraduate research while at Mizzou, and that prompted me to go to Georgia Tech and work on my master's and PhD, where I actually started to work on the sleep data that I'll be presenting to you today. One of the things that I figured out once I finished at Georgia Tech was the fact that I had all of these cool technical tools that I could use, but I really didn't understand the system I was studying, which was the human body and, of course, the human brain. So I decided to do a postdoc at Emory University and work with physicians that are collecting this data from these patients and just really understand what are some of the challenges that they encounter with respect to the systems that they use, what are some of the things they would like to know more about with respect to the signals that they capture, and what are some ways that possibly we could improve the capture of those signals. So yes, if you look at it from a broad standpoint, I have that signal processing expertise, but just looking at it from a more applied approach too, there's this idea of creating these clinical decision support systems. So creating these frameworks that are able to take the data that they collect from the clinic and make these decisions with respect to deciding on whether or not a certain treatment is working, deciding if a patient has a particular disorder, things of that nature. So just to give you an idea of the way the talk will go today, it's very informal, so if you do have like a question or something like that, just be sure to raise your hand and we can discuss as we go along. But generally, I'll have two parts to the talk. The first part will just talk about data science, so that brings in the idea of data mining and what exactly is data science and how am I applying it with respect to this sleep medicine data set. And then giving you some examples of how I've done that. So actually just looking at the data sets that I have, and I will just discuss with you more my thought process as I actually looked at the data and what my approach was. So just to give you this general idea of the framework, data science is what we could consider to be the hub for all of these traditional disciplines such as statistics, mathematics, you have pattern recognition, machine learning. Of course, there's this domain expertise where you actually have the neuroscience area, the sleep medicine field. And data science is beautiful because it brings not only these disciplines, but of course people from these disciplines together, and you have these working teams, and you have these algorithms that are based upon information from all of these different areas that you can maximize on and create systems that are, like I said before with the clinical decision support system, intelligent systems. So you're basing your decisions on this strong approach to one particular area, but then maximizing it. So just giving you an idea of, well, yes, there's this data science, but how does it actually fit within the scheme of the work that I currently am doing? It is basically a general engineering scheme. You have 5 modules. So there's, of course, the data collection. You have to get the data from somewhere. In my particular case, I've looked at human sleep data. So the data is either collected in a research lab or in a clinic. So the clinic being a place where the physician has patients that are referred or patients that just come into the clinic and they have some type of undiagnosed, maybe sleeping disorder. So that data is collected, and then from there, of course, there has to be this approach where you make sure that the data that you've collected is actually the data of interest. And you can't just assume that the data that you collect is what one might say clean. So you have to have some pre-processing approach to make sure that you don't send information to the algorithm or to the system that is erroneous information. So we need to make sure that the signals that we're looking at actually did come from the body, from the brain, things of that nature. Now, there's the third module is looking at the feature extraction and selection. So you have to find a way to take these qualitative descriptions of these signals that the physician sees and actually, take it and put it into a quantitative framework, right? So there has to be some way to translate something that is somewhat subjective into an objective framework. So, of course, now you have these quantitative approaches, but which one do you select? So that is the feature selection. So maybe you have, 50 or 60 different types of ways that you can translate this information, but for each particular case that you're interested in, maybe you don't need to use all 50. So there needs to be some intelligent way to select for what you need to do. Computational costs can be expensive, and if you can reduce that, then you can most likely create a framework that can be operated in a real-time format. So the other part of it is, of course, the classification approach. So now you have this way of translating the data. What decisions do you make? What is the final outcome from this particular mining process? And then there's also the medical expertise, which I include as module number 5, which is more of like a feedback. So this is something where it's more on a consultation basis. So I have medical expertise on there, but there's also that technological expertise too, right? So it's, myself and the physician sitting together, having this conversation about this is the data, is it biologically feasible? What is it telling us about the system itself? How can we improve the way we capture the data? How can we improve the way that we define the way that this data is translated from, this subjective to objective metrics. And so basically, these 5 modules consist of my research, and today I'll focus more so on modules 2, 3, and 4, but I will talk a little bit briefly about module number 1, just to give you an idea of the data itself. And the way that my research contribution is allocated is such that it's about 70% spent on this, computational side. So modules 2, 3, and 4, and then modules 1 and 5, it's more like 30% of my time is spent working in those areas. So the data itself, what does it look like? The clinical terms are called polysomnogram. So poly meaning many, and the somno meaning sleep. So many signals captured while the person is asleep. And so what exactly are we looking at? We're looking at brain activity, we're looking at respiratory, we're looking at heart rate, things of that nature. But of course, there's this decision of what is the information that is of most importance. And I've found in my work, which I'll explain more about the particular cases, that the eye movement activity, which is the electrooculogram, the muscle activity, electromyogram, and the brain activity, electroencephalogram, were the signals that were of interest. So on the left-hand side of the slide, you can see that there are many different signals collected simultaneously over the night, and the subject is able to be observed in these different levels of awake versus sleep and different states of sleep. So just wanted to give you an idea of what are we actually measuring from the body when we look at these signals. So on the engineering side of it, we can look at, well, we're measuring electrical fields, right? We're measuring how these fields are changing with respect to the body. So we assumed with respect to the electrooculogram that there is some electrical field between the retina and the cornea. And as this electrical field changes in positions, we're able to measure that change, and that is what we consider to be the electrooculogram. So if you can look on the right-hand side of the slide, you can see the changes of that electrical field between the cornea and the retina, and how you have a reduction, and you have almost like a baseline and then an increase with respect to going from the left-hand side to the right-hand side. So what are we measuring when we look at the electroencephalogram? So the electroencephalogram is actually, in this particular case, we can consider it to be a measure of the networks of these particular neurons in the brain. So we're not actually doing anything invasive. This is a sleep study, so it's quite difficult to convince someone to do any type of invasive procedure when they're just going to be observed for their sleep. So we have to find ways to be able to still collect the data and to be as non-intrusive as possible. So looking on the left-hand side, you can see that there's this scalp plot, and you have these different letters there. So they have different points on that particular plot where the letters look different. And so that actually is what we consider to be a map. So it's a map of where we're placing, if you can think of it in an electrical engineering sense, our sensors. So our sensors are these scalp electrodes, and what we're doing is we're collecting the activity of these neurons. So it's not one neuron, it's a group of neurons, and how they are excited with respect to some physical state of the body. So you can see on the bottom part of the left-hand slide that you have the group of neurons, and they are excited or inhibited with respect to some type of biological change. And that electrical field, that energy is actually picked up by the electrode. And so that's what we're measuring. And I'll talk more about this as I go on into the talk. But as you can see, that you have to capture this signal as it goes through the skull, as it goes through the tissue. And so there can be some issues with respect to actually picking up the signal itself, so you can get some artifacts. So this system was developed in the 1960s by Rechtschaffen and Kells. And so it allows us to actually work with respect to different labs and know, okay, well, if I'm looking at this particular data set, I know where it's located at on the body. And so the way that it's interpreted is the fact that you have the Fs represent frontal leads and the Cs represent central leads. So these are leads with respect to the actual scalp. And P is parietal, O is occipital. So occipital being the back of the head and frontal being the front of the head. Yes, please.
So are these sensors located specifically enough that you can compare patient to patient, like in California and New Zealand or whatever? Or is there a lot of variation de facto you have to account for?
So generally what they do is they actually do, group people together. But of course, everybody has different characteristics with respect to their skull structure, right? So the way that they use a reference with respect to the person's nasion, so this kind of like bridge part of your nose, and then they do a measurement, which is, that's why they have this 10% and 20%. They do a measurement with respect to those values that they have with respect to that particular person's reference point. So that is how they decide to normalize it with respect to where it's located. Of course, there is going to be some variability because you can't say that each person will be exact in how they place those electrodes. And if they're on the body for a long time, of course, the person could sweat, and in different parts of the experiment, you can have issues with respect to making sure that they stay in place. So, I mean, that could be an issue, but in general, just some of the literature that I've read, they've done tests where they actually had a person, do this setup and then come back a year later, do the same setup, and there were very, very like non-negligible, it's like it didn't even really matter with respect to how different the data was. The data was pretty much very similar. So yeah, that's pretty much the approach that people take, is that these are considered the standards. And from looking at these observations in the clinic, they were able to look at the patient and observe that there were different types of sleep. So they found that there were states where the person had this rapid eye movement, so their eyes would blink while they're sleeping really fast. And then there were points in their sleep where they did not have this rapid eye movement. And so it was broken into these stages of non-rapid eye movement sleep and REM sleep. And so if you look to the right-hand side of the slide, you see this, it's actually like distributions of different signals. So with respect to their particular state and their behavioral state. So if you can think about it along the lines of when you look at the awake signal, it's very fast. The amplitude is very low. So that in so many ways makes sense, right? These networks are exciting. They're working very fast. So you see that the signal is oscillating a lot. So then we go into this area of looking into like stage 1 sleep, where you're a little bit drowsy, and you see the signal, it starts to slow down a little bit. So when you look at that second signal that's on the right-hand side of the slide, you can see that there's also some higher amplitude. So that makes sense, because now these networks, since you're a little drowsy, they're beginning to slow, right? They're beginning to be less excited. And as you go deeper into these different stages of sleep, so if you can kind of think about when you're drowsy and someone makes a sudden movement sitting next to you, then you would wake up. That would be stage 1 sleep. And stage 2 sleep would be more so along the lines of you could have that type of movement, but you wouldn't wake up unless maybe someone called your name. And so you could see as we move from stage 2 sleep to stage 3, that this slowing of the signal gets even bigger, and you see less of these oscillations. And so slow wave sleep is considered this deep level of sleep, and of course, if someone called your name and things of that nature, you generally wouldn't be able to wake up. And so now we have REM sleep, which I spoke about earlier, these rapid eye movements during the sleep process. And there's some aspects of REM sleep that are very similar to the wake signal, and so it's called paradoxal sleep. So are there some things that you can see with respect to the wake signal and the REM sleep signal that would make you see why they call it paradoxal sleep? So is there anything that someone could say regarding the similarity of these particular signals with awake and REM sleep? Somebody want to like just throw out some stuff that they see that's similar? Yeah, so they seem to be around the same amplitude, right? They seem to be, around the same oscillation. So one of the things is, how do we find ways to, classify between these two different types of signals if we don't have video or something like that? So that's why it's really important to make sure that we can have these quantitative approaches where we're looking at the signals in a way that we can, find these different states. So one of the aspects of REM sleep beyond just the rapid eye movement is the fact that there's this lack of muscle movement with respect to REM sleep. And we're not exactly sure why that is, but it seems like it might be some type of system where it makes sure to keep you safe when you're dreaming, and so maybe you won't act out your dreams and hurt yourself or your bed partner. But of course, that's the assumption. There could be other reasons for it too. But we know that when the system goes awry, that individuals tend to have this activity, right, with respect to REM sleep. So we can use this understanding of, not only just the signal itself, but how the signals with respect to other aspects, not just the brain activity, but the muscle activity, eye activity, how they all come together to help us understand these different states. And so the electromyogram is basically, it's just what it is it's a measure of the muscle excitation that you get from the central nervous system or the peripheral nervous system. So these signals are being sent to the muscles, and you get either this extension or contraction. So it's what we can consider almost as a secondary measure of brain activity, right? So now that you have an understanding of the signals that we're actually looking at with respect to this data mining approach of the human sleep-wake cycle, let's look at some of the issues with respect to collecting the data. So I'm going to show you some things that I had to look out for with respect to artifacts that you can find in the signal and issues that you can have if you just take the raw data set and process it. So, excuse me. So in this particular slide, I wanted to give you some examples of some of the common artifacts that you can find with respect to looking at these, sleep studies. And so one of the major things that is a very strong contributor to the fact that we have these issues is the fact that, of course, yes, these signals are collected all at the same time. So you can actually have some of the brain activity and the eye activity. There's a little bit of interference just because those electrodes are placed in close proximity to each other, right? So the first example, if you look at the second signal on the slide, you'll see that there are these huge dips in the signal. And that doesn't represent any type of brain activity. That just represents that the person's eyes are moving in this slow kind of rolling format. And we want to make sure that we don't feed that to the system to say, you know, this is something that represents a brain state when it really doesn't. So we have to look out for that. Another contributor, if you look at the third signal from the top, is the fact that you sometimes can pick up this muscle activity. So we also collect information from the chin. And if the person is maybe, you know, grinding their teeth in their sleep or something like that, then you can see like you have this really, really fast activity. If you look at the latter end of this signal, that is not necessarily representative of the person having this excited brain state. It's just representative of the fact that this person is grinding their teeth. And, yeah.
So these signals here are all EEGs, they're all brain things, but then there's like a tooth grinding happening, and that's how it affects the clean signal. Okay.
Yeah, and so that's a great question. So the question you asked is what signal is being interfered with? So in this particular case, we have an EEG signal, and there's interference from, you know, some eye movement, some interference from some muscle activity, and of course it can go any other type of way too. So, but just for this particular case, I just stuck with the brain activity. And yes, and the last one is just a case where the person is blinking and there is some activity from that blink that is actually interfering with the brain activity. So, you know, there are different ways you can think about, you know, filtering out this information. You can use different types of high pass and low pass filters and things of that nature. But then you have to decide on how to set those filter coefficients, and, you know, do those filter coefficients change with respect to patient, which was a question asked earlier with respect to, you know, how do we have this almost like normalization of our data set. So the way I looked at it is I searched the literature and looked at what are some of the approaches that people are using that have signals that are similar to mine. And so one of the things I found in the brain-computer interaction arena is the fact that they actually look at this signal source separation technique. So they have this idea that with this signal source separation, there's this embedded noise with the signal. So they're almost like in the same plane. And so the goal is you want to try to transform the data such that you can make sure that you get the highest signal to noise ratio, right? So you have to find some way to make sure that you can take that noise and extract it to the best of your abilities. And I found that the generalized singular value decomposition algorithm is a linear algebra approach at decomposing matrices that allows you to pretty much find these eigenvectors that give you this orthogonal representation of your signal and your noise. And so if you look on the left-hand side, you can see that idea of the signal and having the embedded aspect of the noise. And on the right-hand side, this kind of gives you a visual of what I mean by transforming the data and putting it in a different plane so that you can have this separation of the signal and the noise. So the great aspect about doing this is the fact that we can now not just look at making these high pass and low pass filters for specific types of artifacts. We can actually just focus on making this idea of what our noise vector is as optimal as possible, and then using that for, you know, all of our data set. And so it actually on the end of the process creates less work than trying to create, you know, 7 or 8 or 9 different types of filters for all of the different types of artifacts. So I wanted to give you an example of, you know, what does some of the data look like when it has these, you know, artifacts in it. So before I gave you an example which was more just like a toy example, but this here is actually, you know, real data. And so, you know, what does it look like when it's contaminated? So here I have 29 channels, you know, from the Polysomnogram, and I actually have included in here, you know, some other channels from, if you see the top signal, it's the EKG and some muscle activity. But the main example here is with respect to some eye blink activity that is actually interfering with the EEG, and then there is some muscle activity that's also interfering with the EEG. So just to go in line with the examples I gave earlier. So just to kind of get an idea of, what output could we get from using this, GSVD approach. On the left-hand side, you can see the original data set. And on the right-hand side, you'll see the data set post-GSVD. So post using this transformation of the data. And so you look into the red rectangle, and you can see that there's a lot of this muscle activity from, say, probably the right leg or left leg. And it is interfering with these frontal leads, so these FP2, F3, F4, which is at the lower part of the slide. And post-GSVD, we're actually able to get this better representation of the underlying frontal activity from the brain, right? So one of the contributions I did in this particular work was to try and find a way to do it in an automated fashion. The way that this approach, GSVD, is used in BCI before was where the person actually had to sit and look at the different eigenvectors and say, oh, okay, I don't want to include this one, I want to include that one. This one resembles more so the noise I'm interested in, that one doesn't. And so I decided, instead of having the person sit there and do that, of course the physician is not going to have time to do that, or maybe they won't even feel comfortable sitting down and looking at those particular components. I thought that it would be great to just use the cross-correlation function. So cross-correlation gives you this idea of the similarity between signals, right? So you can look at the similarity between what you would consider to be an artifact and what you want to be your signal. In this case, I took the EEG, so the brain activity, as the signal of interest, and made sure that whenever that matched to a certain arbitrary value, in my particular case, I did 60%. So if it was 60% or more correlated with the other signals, that were not EEG, then I consider that to be artifact. And so then you use the GSVD. So it's not using this GSVD on each aspect of the data. It's only using it when you have this signal interference. So I use a 1 second moving window, non-overlapping, to do that. And this is another example of, say, an eye blink that is interfering with the temporal leads of the EEG. And so you can see with using the GSVD that you can see more of the underlying information from the temporal leads. So one of the questions that can come up with this is the idea of, how do we know that we're also not getting rid of the EEG that we're interested in? It's a good question, right? So yeah, we don't know for sure. So you have to find ways to try and assess that, right? So one of the things that we can do in order to do that is to come up with these metrics where we look at the data pre and post. One of the things I looked at was the correlation value pre and post the GSVD. But other things that I will look at going forward will include looking at like the power spectrum, pre and post, and then also making sure that we can understand the power spectrum of signals that have no artifact, completely, and then using that as more so of a baseline. But the great thing about it is it's not just a visual change. There's also this, quantitative way that we can express this reduction in this artifact. And the fact that we can use something that has this way of dealing with mixed artifacts is a great thing. So again, using the filter approach is great, but then you would have to decide when to use the filters, how many filters, a lot of computational costs. So this actually allows us to find a way to process these mixed artifacts and to also have something that's computationally feasible, right? So now we have this data set that we can consider to be, clean. So we can now run these features on that data set and find ways to actually, extract this information that we're looking for and start to do this, deeper analysis of making these particular decisions. So if you can, direct your attention to module number 3, you'll see that example of what I talked about earlier with respect to, using this 1 second window to, calculate this information. It's a moving window, and you can calculate, in this particular case, we consider a feature to be very similar to if you were describing a friend and you said, I want to describe this friend, and I will say that I will describe this friend by respect to their height or what color clothes they have on. So it's pretty much a descriptor of the signal. So in this particular case, we can say that, just for example, the descriptor for this particular signal could be, say, the amplitude. So you take the average amplitude value and you calculate it with the moving window, and you get some, feature matrix or feature vector. And from there, you can use that. In this particular case, it'll be in a training format. So you have someone who has labeled this data to let you know what particular window is what particular, say, sleep stage. And then you can use that to actually create your system that you would have to represent, quantitatively what we're dealing with respect to those states. So the idea is you come up with this feature library, which could be many different types of, quantitative, representations, functions, things of that nature of the signal. So it could be in the time domain, it could be in the information domain, the statistical domain, the frequency domain, which was some things that we looked at earlier with respect to the oscillations, the slow, the fast, and then the wavelet domain. So we can also look at not just looking at this Fourier approach, but also looking at other types of basis functions. So just to kind of give you an idea of what I mean with respect to some of the cases with respect to the types of data sets that we're looking at. I have, in this particular case, looked at 2 particular projects. So one project deals with this idiopathic hypersomnia. So there's this excessive sleepiness, and the person can, sleep for over 11 hours and still wake up and feel unrefreshed, or they have this inability to wake up. And so, of course, you can have these concentration problems. And one of the things that's interesting in this particular approach is how do we use those features in order to, capture this person's level of vigilance, and then capture it enough such that we will know if a treatment is working or not working. So yes, the patient can say, I don't feel better, or I do feel better, but how do we quantify that? And so the other aspect that I look at within my research is looking at neurodegenerative disorders. So looking at pathologies where there is some destruction of the brain cells that we looked at earlier, and how that impacts how those networks are represented with respect to those signaling. In this particular case, I don't necessarily look at the brain activity, I look at the muscle activity, because I'm looking at cases where the patients have Parkinson's disease, which is a movement disorder. So there's this slowness of movement, there's inabilities to do voluntary movements. And so we want to look at, how does the EMG change with respect to the progression of this particular illness? And maybe we can use this as a marker to tell us, okay, this person has this tendency to fall into the category of being someone that would develop, Parkinson's disease. So one of the great things that we found is the fact that people that have this, movement disorder issue with Parkinson's disease, some of their issues with respect to pathology show up in their sleep before they show up with respect to their movement. So, looking at these particular approaches are really great because it can give us a whole big picture with respect to looking at the pathology, and being able to intervene at a certain time to help the patient. One of the other categories I look at is rapid eye movement behavior disorder. And so in this particular case, we looked at earlier how, in the healthy case, there is no movement during this REM sleep. But in the unhealthy case, for some reason, that signaling that's sent to the muscles to say, yes, no movement, is interrupted. And that causes a lot of different, movements to occur while the person is in their REM sleep, which, they act out their dreams, they can hurt themselves, their bed partner. And so the goal is to, better understand this illness, especially since 50 to 60% of the patients that have this REM behavior disorder go on to develop Parkinson's disease. So there could be some relationship there with respect to this, movement fluctuations and how they are going on in developing these neurodegenerative conditions. So these are the types of cases that I look at with respect to, my data sets. And so some of the features that I look at with respect to understanding the data sets, including from the time domain, I can look at the curve length. So I can look at, like when we looked earlier, how we saw that the signal with respect to going into these deeper stages of sleep, that the signal, the amplitude was a lot higher and the signal itself slowed down. So we can look at the curve length. We can look at these inflection points. How often does the signal peak and how often does it trough? We can look at the information domain. So Shannon's entropy. So how predictable is the signal with respect to particular sleep states, right? So this is just a very basic example where you have, a basic function that's oscillating very distinct pattern on the left-hand side. And on the right-hand side, it's more random. So the right-hand side would have a higher Shannon's entropy than, say, the left-hand side. And then you have also this idea of looking at the peakiness of the signal. So, this is kurtosis, how does it fit a certain type of distribution? So you could say, does it fit like a normal distribution, things of that nature. So in this case, you can see the red signal is very peaked, so it'd be a high kurtosis, and then the square doesn't have as much of a peak, so low kurtosis. And of course, we look at these signals in relation to the particular type of events of interest. So just to kind of get deeper into the data here, one of the cases that I looked at with respect to the hypersomnia case was looking at a way to see if a pharmaceutical intervention was actually working with respect to changing the patient's vigilance. So this can help out with understanding dosages, right? So making sure, are we giving the patient, too much? Are we giving them too little? So what we did is we took some data while the patient was going through a vigilance task, and we had them actually, recording their EEG. So we record their brain activity where they're supposed to be awake, and they have no type of intervention, no pharma treatment. And then we give them a pharma treatment, and we have them do the same vigilance task. And we look at their brain activity, after that. So here we just see, in this particular case, the take-home is the fact that you can see that there are some increases in the higher frequencies of the brain, which, it could tell us that there's some, increase in some excitation with respect to this particular treatment, and just giving us some idea of, how that dosage is actually impacting the patient's vigilance, beyond just giving them a subjective survey to say, do you feel awake or do you not feel awake? We need to know quantitatively, how is that awake state changing? How's that vigilance changing? So this is just an example of one feature. And the next thing, again, is now having these features, there's an idea of looking at the classification of it. So how do we decide to say the person is in a healthy state or a non-healthy state? Does their data fall in particular sleep stage, non-REM sleep or REM sleep? So I just used in this particular case a K-nearest neighbor classifier. It's what is considered to be a supervised classifier. So you have some data, you have someone label it, so you have a ground, or you could say a baseline understanding of what the data is supposed to look like. And you use that as your standard approach to say, when I have some new information, then wherever that new information falls with respect to the information I already know is how I'm going to label that information that I don't know anything about. So in this particular case, it's just using what you consider to be just a distance function. So how far or how close are the data point that you don't know anything about to the data that you do know something about. So in this particular case, you can see that you have this, the red dots. In this case, we could say they're stage 2 sleep, and the black dots are REM sleep. And so we could say that the distribution falls somewhat like it is on the right-hand side of the slide. The left-hand side of the slide, it just shows you the distance equation itself. So if you were just to take a guess as to the point that has the question mark, and you would say, well, based upon how close, whatever class that I'm looking at is to that unknown data point, what would you say you would place this particular data point in? Would you say you would place it in the stage 2 red dot class, or would you place it in the black dot REM class? And we're actually saying that we will place this data and label it with respect to how close it is to a particular class of data that we have information on. So how many people would say that the question mark, you would put that data in the REM class? Okay, how many in stage 2? All right, stage 2, yeah. So it's very straightforward, as you can see. But one of the issues with this particular approach is the fact that, yes, you have to have a certain amount of data to do that. So as I move forward, I'm also looking at ways that are unsupervised, but this is just an approach to just say, what are some of the features that are of interest? So we can at least have some features, and then once we have that, we can go look at some more unsupervised approaches. So my time is running a little bit low, so I don't want to spend too much time on some of these slides, but I'll just give you like a little brief information about it, and then I can get to the last point that I wanted to talk about, which was the neurodegenerative disorder case. But basically, we have all these different features. How do we decide which one to use? So we have these great feature selection algorithms that come from machine learning. And these are just examples. They have sequential forward feature selection. So you start off with this idea of initializing, where you would decide to, say, select a feature. And then from there, of course, this is again based on this training data, you would add different features, and you would try to maximize some type of equation with respect to your training data, so that you would come out with some feature subset that best fits with respect to that training and testing data. And so the sequential backward feature selection, very similar. Instead of just starting with one feature, you actually start with all your features, and then you drop features, so that way you're not processing information that's redundant, right? You don't want to use up your computational resources for something that is giving you the same information that you already have. So in this particular case, this is just an example of trying to figure out from all of these different features, which ones will work in a particular population. So I looked at features for a population of data sets that I have for Parkinson's disease and a population that I have for normals. And the goal was to just make sure that I could mimic the same type of scoring for the sleep stages that the physician did. So in this case, one of the take-home points from this slide is the fact that you do get this surface plot, so you get this terrain, and the goal is to make sure that you can maximize, get to the highest point on that terrain but then also make sure that you're not, over using your resources. So one of the things that we could do is see from this particular slide that we could take, for example, the normal controls case, how maybe just using in this particular case, say, what is it, 2, 3, 7 features and maybe 5 nearest neighbors will be okay instead of just using the 20. So if you look at the first arrow, that's closer to the front of the screen, you have less nearest neighbors versus you would go to more in depth into the screen, you would have more nearest neighbors. So it's just saying, how can I intelligently select the parameters, right, for my particular, model. And the last example I wanted to go over was respect to looking at this, this is now moving just from looking at these state changes. So like, instead of just looking at, is this person asleep, what type of sleep? Looking at this microarchitecture, so looking at things within the sleep process that can come out such that, this might be interesting too, not just knowing if the person is in a certain type of sleep, but what's going on while they're sleeping. In this particular case, just working with a physician, they found that they saw these little like phasic twitches that came up that were very, very high in density for patients that have this REM behavior disorder. And compared to controls, you didn't see it that often. And so there was this idea of maybe we could use that as a marker to let us know that this person is developing RBD, which could also go on to develop Parkinson's disease. So you can see here the incentive for being able to track these different events. But the problem is they're short, they're tedious to mark, and so who's going to sit there and mark these events every 1 second to say they occur or they don't occur? So this is a great way to actually use these techniques that we have in data science to be able to track this information. And then it's also, like I mentioned earlier, not just saying it exists there, it doesn't exist, but also looking at things that, say, the physician wouldn't be able to analyze, because we can also look at the intensity of these twitches, we can look at the interval between them and things of that nature. So just in general, we use the wavelets in this particular approach because the wavelets, they actually look very similar to the data set itself. And again, we're just looking at the data, comparing it to this standard visual approach. And we saw that we were able to detect these little twitches, so this phasic activity with both types of wavelets. So one didn't outperform the other. And we were also able to detect the non-phasic events. So just some of the contributions are the fact that we're able to create these approaches that not only just processing the data, but doing it in an automated way, not only just detecting events, but being able to expand upon that detection of events, and then also being able to make characterizations about the data set that are not at this point feasible with respect to visual analysis. And so these are just some of the individuals that helped me with the work, and I hope I didn't put you to sleep. Thank you.
So actually, we're at the end of our time, but I've scheduled about 10 or 15 minutes before our next meeting, so we can just go out in the lobby if you have questions. So thank you, and come talk to us.